For those who don't know me, my name is Dr. Kat Sebastian, and I'm head of the Mental Health Evidence Team at the Wellcome Trust. It's a pleasure to welcome you here to this symposium on animal models for mental health research. As many of you know, around about 3 years ago, Wellcome committed to having mental health as a key priority area. And as my role as head of evidence for mental health is to ensure that we fund research which helps us to better understand how anxiety, depression, and psychosis develop and also how they resolve. We owe it a key part of both. So this means that we've got a key interest in funding mechanistic research which helps us to better understand the processes which underpin mental health problems. Neuroscience is a key part of that, and animal models also. And as a funder, Wellcome supports the use of animal models as long as such research can show that it is scientifically, legally, and ethically justified. Last year, we ran a funding call aimed at using computational methods to improve our understanding of mental health symptoms such as anhedonia or hallucinations. A scoping report that we commissioned noted that current animal models have critical shortcomings in how they recapitulate key features of anxiety, depression, and psychosis. We owe it to both potential human beneficiaries of mental health research and the animals that we involve in our work to develop animal models of mental health symptoms and conditions that are more valid reflections of the symptoms that clinicians are aiming to treat. The talks that we'll hear today all touch on this theme in different ways, and all are at the forefront of improving translational work through better use of animal models. Our first speaker, Professor Geoffrey Daly from the University of Cambridge, will talk about bridging the gap between cells and the clinic in understanding stress-related mental health conditions. A second challenge in animal models research in mental health is thinking about how to integrate the voice of lived experience into mechanistic research with animal models, which can sometimes feel quite far removed from the symptoms that we're aiming to treat and can sometimes potentially seem reductionist. Our lived experience team at Wellcome, led by Dr. Kate Martin, has been thinking about this a lot, and we're lucky to have lived experience advocate and MQ ambassador James Downs to talk about this in more detail. Another challenge that we've identified in our work at Wellcome is that it's not currently easy as a mental health researcher to find a definitive source that maps out existing animal models and how they link to particular symptoms and conditions so that you can select the best model to use in your research. My colleague, Dr. Niall Boyce, who's here in the audience today, and head of the knowledge and measurement team at Wellcome, has commissioned what we're calling a library of animal models with a team at Monash University recently appointed to conduct this work. We'll hear more about this from Dr. Rachel Hill in her talk. And finally, we'll hear from Dr. Georgia Salanti from the University of Bern, joining us online, who will talk about her work on the Galanos project, another Wellcome-funded project that's looking at how we can use the outputs of both animal and human research for evidence synthesis to pinpoint new research priorities. Before I hand over to Jeff for our first talk, I just wanted to highlight that for the structure of this symposium, what we're going to do is set aside a bit of time at the end so that we can have a panel discussion with all our speakers so we can discuss some of the broader issues as a group. And there'll also be a chance for one or two specific questions at the end of each speaker's talk. So during the session, please do think of questions that you might want to ask the whole panel at the end, and hopefully we can get a good discussion going. Without further ado, I'll hand over to Professor Geoff Daly from the University of Cambridge.
Thank you, Kat. I've been told I've got 17 minutes. Now, 17 minutes is a tricky time, so I will try to bring it in on time. Okay, what I'm going to do in this talk is give a sense of some of the ideas that we're developing around stress-related research. This idea of a stress memory, something which is persistent, you encode early, and potentially can interact with further stress to increase the probability of adverse outcomes. And I'll focus on anxiety, depression, but pointing out the value of animal research and how we can build translational bridges with humans and human population and clinical groups. And I always think it's important with research and research talks is to say, well, why are we doing this in the first place? It costs hundreds of millions, billions of pounds on stress-related disorders. We can think of depression, anxiety, addiction. We can think of GP appointments. The amount of energy, cost, time, resource on these. It's hugely important that we understand mechanism. And thinking of mechanism, we have medications for depression called SSRIs. We know the primary target, the SERT, the serotonin transporter, but mysteriously, after all of these decades, we still really don't have a clear notion of how these drugs work. And in fact, 30% of patients prescribed SSRIs show treatment refractory outcomes. And so we need to develop better medication. And of course, we have some new kids on the block. We have derivatives of ketamine, S-ketamine, the nasal spray, fast-acting. We have magic mushrooms. Well, we don't have magic mushrooms to treat depression just yet, but there are some interesting active ingredients whereby we could begin to think about how we might change the way we think about the treatment of these disorders. Slight theory, detour here, Hans Seeley, pioneer of stress research. The idea about how we move from alarm resistance, the helpful aspects of how we adapt to stress, well, that's fine, but we want to stay in those first two stages. Harmful stress outcomes occur when we get into that final stage, when we become overwhelmed, stress is intense, uncontrollable. So if we're thinking of medications and how we build animal models, we need to be thinking of ways of strengthening resilience and keeping us away from that third stage called exhaustion. Amy Arnstead came up with the idea that there is a balance between bottom-up amygdala striatal-based mechanisms and a top-down prefrontal process which acts to balance and neutralize the amygdala striatal habitual systems. Exerting effective top-down control will be a theme of this talk. How can we strengthen the way in which we control these subcortical habitual systems? And the point I will make is there is something special about early life stress which interacts with additional stresses later in life. We know in the maternal environment how both nutrition, environmental stresses can interact to change the neurodevelopmental outcomes of developing babies. And there's some classic imaging studies. This was Edmund Sanuga-Bark looking at Romanian orphans. This was a very severe form of deprivation, but we can see the example here, enlarged volume of the amygdala in these Romanian orphans. And in Cambridge, what we use is a much more subtle procedure. This is called the repeated maternal separation procedure. I say it's a relatively mild, I'd say it's disruptive to the social interaction between the dam, the maternal aspect, and the offspring. And what we do here is we separate the offspring from postnatal day 5 right through to the beginning of adolescence, which is postnatal day 19 in our hands. And then what we do, we allow the animals to grow up to adults, and we test these adults on various touchscreen tasks, behavioural assays, which match the equivalent tasks in humans. We know that this period of maternal offspring interaction is important. Mike Meany showed that mums, good mums, and lick a lot and groom their offspring a lot, develop healthy levels of the GR receptor. This is the receptor upon which cortisol acts. By contrast, poor mums do the opposite, and you have this epigenetic modification of the promoter region. And this hypermethylation means fewer receptors, and the stress response continues because the stress response through negative feedback is not switched off. Ethan Dusha was a PhD student, just finished recently in my group, used high-resolution imaging, MR imaging, using the maternal separation stress procedure. And it was a touchscreen task, and it was a couple of important observations. The first is that when animals do a touchscreen task, there's a choice, right or wrong, and asked to switch to look at flexibility, we found that the animals would learn by biasing to negative feedback. So they're incredibly sensitive to the negative feedback. I think that's an important lesson for how we might think about developing tasks. But crucially, Ethan also discovered that the amygdala bilaterally was enlarged in these maternally separated animals. And there's obviously a correlation then with what we saw earlier with a more severe case of deprivation. And just to extend the imaging story just a little further, Catherine Zusdorf, another PhD student in my group, used a seed-based functional MRI approach. And the seed was placed in the amygdala, and we're here looking at the relationship, the correlation in the bold response extracted from the amygdala and frontal cortical regions. And actually what Katharina showed was that there is underactivity, reduced connectivity between the frontal lobe regions and the amygdala. And already perhaps there's a clue there that there is a change in the relationship between the frontal cortical regions and the amygdala in animals which have experienced an early life stressor. And we can see parallels in humans with depression. If we look at similar or analogous regions in humans and use a similar approach, we can see evidence of reduced connectivity strength between these frontal regions, anterior cingulate cortex, insular cortex, and the amygdala. All right, so that leads me nicely into this very exciting new grant. It's funded by Wellcome. Camilla Nord, Becky Lawson, there's a whole team there, Steph Archer, all involved working both in humans and rodents using the same tasks. And the idea of this grant is to look at uncertainty. Uncertainty is a powerful driver for anxiety, and we can develop anxiety and a misestimation of uncertain information if we don't firstly have a belief system which is correct, and we have a learning system which is also aberrant. We learn too fast, we learn too slow in a certain context. So the whole idea of this grant is to what we do called uncertainty fingerprinting. We're using computational approaches to model latent variables on a number of tasks, touchscreen tasks, basically the same in humans and rodents. One of the tasks is looking at flexibility on a probabilistic reversal learning where there's spurious feedback, and an animal has to track the most optimal rewarding outcomes and decide to switch or not in those conditions. And the exciting aspect for me about this grant is it's reverse engineering drugs which we already have in the clinic. So SSRIs, as I've already told you, we know the primary target in the brain upon which they act, but we have very little information about the effects clinically and how it remediates depression. And there's so much that we simply don't know. One large component of the grant is looking at early life stress as a special form of stress where the plasticity that stress can exert these persistent changes in the developing nervous system are so important. What we're looking at are tasks and modelling belief states when there's uncertain or volatile information. We're looking at the encoding of this information, and essentially what we're trying to estimate is uncertainty misestimation. And by looking at SSRIs and cognitive behavioural therapy, we can begin to ask a different question about these drugs. How do they change the way in which people decide how things work, their belief states? How do we update information based on new information? On the rodent side, which my lab works, it's mechanistic. We're developing grab sensors to measure noradrenaline, 5-HT, to understand trial by trial how an animal learns. Does it learn to positive feedback or to negative feedback? So that's the grant. We're underway. The tasks are developed and decided upon. We're early stages. But I thought what I would do, I've got just a few minutes, is a departure on a study which came about. We followed a lead for a while, and we decided, actually, this is quite good, and we're going to stick with this for a bit longer. Essentially, we did a bulk RNA sequencing experiment, looking at the prefrontal cortex. Remember, this is the top-down region exerting control over the subcortical amygdala and striatum. Bulk RNA, so we're agnostic to the cell type. We just look at all the RNA transcripts in the prefrontal cortex of maternally separated animals. And the question we've been grappling with is whether early life stress alone is sufficient and necessary for depressogenic phenotypes, Or do you need to add something else, more stress? This is the classic stress diathesis model. In other words, you have a genetic predisposition, or there may be early life trauma, and coupled with further stress can alter the probability of whether you develop and express the clinical phenotype. And with this RNA sequencing experiment, we discovered, using gene set enrichment analysis, that all of the genes which were upregulated, both in males and female maternally separated animals, sat in the electron transport chain, the oxidative phosphorylation systems. This form of cellular respiration is incredibly important for how these prefrontal networks organize control, top-down control. And through a really important recent collaboration, we're now looking at dynamic measures of cellular respiration. This is with Andrew Murray and his team, PDN in Cambridge, and using different substrates like pyruvate, ADP, glutamate, you can look at the different complexes to see oxygen consumption. Which of those protein complexes are responsible for generating ATP, the final outcome, energy currency in the CNS. And Carmen Sandy in Switzerland has done some seminal work in this field. The stressor here was young adult, post-adolescence. And what Carmen discovered was that complex 1, you actually see increased oxygen consumption. So there's an upregulation of this complex. This work was done the day after the end of the stress. What we've done is something a bit different. We've actually looked at this oxygen consumption 9 months after the early maternal separation stress, 9 months. And what you can see in this figure is there's the control groups. We're looking here at oxygen consumption, control animals here, and adults that experienced a second stress. Here's the maternally separated animals, and you can see there's a significant increase in oxygen consumption. There's increased oxidative capacity, but look what happens when you expose the animals to a second stressor. This is an inescapable foot shock. It uncouples, and you get a reduced consumption of oxygen. And the theory that we're working on here is that perhaps the early life stress is building resilience, but there is a cost, a trade-off to that early resilience. In fact, you may actually show increased vulnerability to additional stresses that you experience. And the reason for that is that some of the byproducts of the oxidative phosphorylation can feed through the reactive oxidative species chain. And we've recently discovered that actually if you just look straight after the end of, or postnatal day 20, the end of the maternal separation procedure, particularly in males, there is an uncoupling, reduced coupling efficiency. This is a classic phenotype that when you're experiencing stress, for every ounce of oxygen that you consume, less ATP is formed. The result of that is increased biogenesis. You make more mitochondria to compensate. So you can see already that there is evidence of increased biogenesis, a form of resilience, establishing it, if you like. So what are we going to do in treatment in the last 2 minutes? How are we going to do something useful here? Well, it turns out the 5-HT2A receptor is upstream target controlling this bioenergetic pathway. And the healthy stuff is that if the 5-HT2A receptor is activated, it can run through this G protein coupled receptor, increase biogenesis, increases oxidative capacity, it's neuroprotective, and a reduction in the ROS signals, and more ATP, all good. We decided then to focus on that 5-HT2A receptor, and we just carried out a recent study where we had control and maternally separated animals. They went for the early life stress procedure there in green. For those postnatal day 20 to 79, it was normal husbandry. They then were exposed to a second stress during adulthood. And then we treated these animals either with saline, citalopram, twice a day for 1 week, or citalopram coupled with a 5-HT2A receptor antagonist. This was trying to separate out what the 2A receptor might be doing in this procedure. And at the end, we sacrificed it at 110, and we did respirometry to look at oxidative phosphorylation. Now I'm going to show you some data which is completely against prediction. We predicted that citalopram would just be beneficial. It would just increase oxidative phosphorylation, add to the stress resilience that happened earlier on. Here's the data, and we've got complex 1 and A, complex 2 and B. The control animals, the 3 bars on the left, the maternally separated animals there on the right. Citalopram actually reduced oxidative capacity in a maternally separated animal. The control animals, the only difference there was they didn't have the early stress. So something is persistent and interacting with the additional stress, and citalopram to actually compromise oxidative capacity in these animals. This is a problem we're grappling with now. We're doing gene transcription, we're looking at 5-HT release. There is something responsible for that early effect of citalopram, which is not necessarily helpful. But it brings me back to magic mushrooms, psilocybin. Of course, a lot of interest in the 2A receptor and how we might build stress resilience. And Robin Carhart-Harris and Dave Nutt have had many ideas about how serotonergic agents might work in depression. It's literally penultimate slide, I think, or 2 more. The 2A receptor important cortically, and this is about how we think about beliefs, a reset, if you like, and there's a subcortical 5-HT1A component. So, theoretically, what are we thinking is going on here? Well, you can look across the lifespan of cumulative uncontrollable stress. You could look on this axis on mitochondrial function in the prefrontal cortex, the top-down mechanism. And of course, all stress researchers will know that inverted U-shaped functions are used commonly. And the maternally separated animals, you build resilience up to a point. With further stress, you see an uncoupling and a decrement in the way this perhaps, reputatively, mitochondrial function of the prefrontal cortex exert top-down control. I won't bore you with all those concluding remarks, but I think the point to make here is that early life stress leaves a mark which lasts for months, and it interacts with additional stress. And when we think about how we treat depression and other stress-related disorders, we need to be thinking about that. And there's a whole heap of people there that I need to thank, and for the Wellcome Trust for funding this really exciting grant. Thank you.
Is this on? Thanks, Jeff, for sticking to time and imparting so much so many interesting results in such a short space of time. I think we've got space for 1 or 2 questions.
Thank you so much. Great talk. I was wondering, when you were talking about the fact that chronic stress results in building resilience that has this footprint in mitochondrial function, but that confers increased vulnerability, I was thinking, are the animals, when they are deprived from their mother, undergoing any kind of hypoxia because they scream too much or they don't breathe enough? Is it a physiological adaptation, or is it a functional adaptation instead?
Yeah, I mean, that's a really good question. There are so many possibilities about why that group of animals may become resilient and immunized against additional stresses. I mean, unless you measure these things, I mean, it's an open-ended question, but yeah, we can't rule it out. There may be physiological adaptations. Yeah.
Thank you. Thank you ever so much for the talk, Jeff. I just have a question with regards to you're talking about prefrontal as an entire unit. I just wondered whether you could speak to, in terms of your data, whether there's any difference across different substructures within the prefrontal, and whether that has some importance in terms of resilience or vulnerability.
I mean, yeah, this included, it collapsed both infralimbic, prelimbic, anterior cingulate. It's a medial prefrontal cortex. The subregional analysis would be important. Secondly, cell type specificity. There's some new evidence in humans that the mitochondrial phenotypes are glial rather than neuronal. So there are 2 really important areas that we need to be working on. But yeah.
Thank you.
Yeah.
Great. Thanks. I think we'll leave the specific questions there. And let's welcome James Downs, who's going to talk about his work with lived experience.
Can you hear me all right? Yes. Okay, so these are by far the least scientific slides that you're going to see all conference. My mind is blown already. I'm going to be telling you a bit of a story, and I think it's hopefully going to illuminate some things that some of the science might not help you to see so obviously. So over 20 years ago, I was diagnosed with severe anorexia, and I was quite lucky in a way because I was already in mental health services. I had OCD, anxiety, depression, things that we know relate to eating disorders. So you'd think, great, we have the promise of evidence-based treatment, and I was ideally situated for that early intervention to recover. Unfortunately, my story didn't go the way that I wanted it to, and my life has been significantly impacted by over 25 years of severe mental illness that carries on today. And, you know, that promise of evidence-based treatment, I don't think we know what works for people like me from my story. And I suppose a key question is evidence-based for who, I suppose. And I'm not just talking about the obvious ways in which some people like me might be excluded from current research, thinking eating disorders, over 90% of participants in studies are female. But I'm thinking about, you know, people who are not like me, people from different backgrounds, other identities who might not be included at all. And I'm also talking about the hard science. There's so much that we don't know. It was really reassuring to hear that. In eating disorders, there's so much basic science that we don't know, that we need to uncover. What's going on metabolically? What about the changing food environment, ultra-processed foods, how people migrate between diagnoses like I did, and how some people seem not to. And I think it means, like I said, we don't know what works for who, and we operate then in the dark, and people like me are sort of scrabbling around trying to find pieces of the puzzle to help ourselves. And I suppose there's a risk as well that sometimes clinicians might not acknowledge this, that they're operating in the dark, and we might get the impression as patients that the experts are the people who know everything about this subject, rather than being the people who know the most about this subject at the moment, which is kind of a different thing. So we might think, oh, this should work, and if it's not working, then I'm the one who's to blame or somehow extra stigmatized. And I felt sort of like a misfit. I don't fit the treatment or the models that apply to my conditions, and that's made me feel invalid. And as an invalid, I'm on the outside of these models, and I'm living in that knowledge gap. So that's even before we start to think about co-occurring conditions, which are the norm rather than the exception in severe mental illness, like eating disorders. And in my story, there's chronic illness, depression, anxiety, trauma, personality disorder, dissociation, paranoia, suicidality, myopathic Ehlers-Danlos syndrome, a rare genetic condition, hypermobility, gastroparesis, autism, ADHD, chronic fatigue, etc. It's a web, isn't it? And this is, I suppose, what humans are like and what models perhaps are not like. And I'm thinking, where is the randomized controlled trial or the paper that could be pinged up with the results for me, for somebody with that sort of array of experiences? And who's integrating the evidence from the lab and from clinical research to help me with treatment that will fit well, with my experience, rather than saying perhaps I'm a difficult patient and I don't want to get better. Yeah. So I'm not saying that we don't know a lot already. I think we know about, I'll try and be scientific, we know about serotonin and dopamine and gut-brain interactions, and we have elegant models and complex diagrams. The gaps are being filled in, but I suppose the most complete models only take us so far and aren't capable of relating what we know to the lived realities of people like me who need these models the most. And, what about somebody who experiences a genetic change that they don't even know about as pain? Or they have hunger, but they experience it as chronic feelings of emptiness, or people who have obsessional behaviors who experience them as necessary for survival. They're explained in completely different ways, and that word experience, I think, is key. because human experience doesn't happen in these silos that the studies are done in, and it happens within people. And it's not enough to study people as though, one part, in component parts, they don't relate to one another. And I don't want to be understood as a set of disconnected data points. I think people deserve treatment that addresses the whole picture and need to be understood as whole people. So I've kind of co-opted a quote that's quite famous there about the best model of the human being the human. Is my story, I guess, or people's stories, are they anecdotal or are they evidence? I often get told, oh, well, especially if I'm saying something negative, oh, it's just anecdote, yeah? And I think it's kind of both. I'd say it's a bit of both. And evidence for what? I guess it's not, in the case that I've said, it's not necessarily evidence for positive findings, it's evidence for gaps. Yeah? And gaps in not just sort of what we know, but how we come to know about things and how we, who gets to decide, I suppose. I'm really interested in this term, epistemic justice, and the idea of what it is to be a knower. And I think there's a bit of injustice when somebody says, look, what you're saying to be true about your experience, if you're saying, this for me is to do with survival, or like I've given those examples of interpreting your own experience. If you said, oh, that's not wrong, it's not put in the scientific, that's wrong, it's not in scientific language. Or there's hermeneutic injustice, which is maybe where, you're not deemed to be a reliable narrator of your experiences and somebody else, an expert, has to interpret them for you. And I've alluded to some of the harms of that, like having misexplanations for your experience. I've had all kinds of explanations given to me that just don't fit, and I think in a way they've been professionals saying sort of, not wanting to say I don't know, actually. So I'm going to do a really quick experiment because I know I'm running out of time. I'm going to ask you very quickly, anybody, to shout out what you see in this picture, if anything. Face, okay, anybody see anything else? Somebody playing tenor saxophone with a bit of puff of smoke coming out, yeah. Okay, so there are a few options, and half of you are completely wrong, but I don't know which half yet because I'm yet to decide, yeah. And I think that mental health research sometimes can be a little bit like that. Yeah, who holds the power to decide? Whose interpretation is real? And there are many things that I know to be true about my experience that don't show up on fMRI scans, that aren't in the DSM or the ICD-11, they're not in a data set. And so there's something relational about this, isn't there, between people like me and researchers like you or clinicians. And I think we can all think of relationships where we felt, this is true, and you just don't get it, yeah? And I think that we need to be avoiding that, and I think there's a bit of a double empathy problem there. So how do we do this? Very quickly, co-production, I think, is part of the answer here, and not the box-ticking kind where somebody's in the room who has lived experience and, that sort of tokenism, which I've had too many experiences of, but it's changed a lot in the 10 years that I've been doing this kind of work. I think it's really about the sharing the power and to have that alongsideness in, look, let's look at this thing together from our differing perspectives and create a whole new perspective. There are lots of benefits there on the slide that I think that co-production can bring, although I'll acknowledge we're in an infancy with this kind of method. So great, but there are lots of ethical challenges as well, and I think that co-production can be really disruptive. It's about, what are you going to say to the funders when you don't know what the people with lived experience are going to do yet because you haven't got to that bit yet, and you just say to them, oh, they'll decide sometime down the line, are they going to give you the money? And what about when you're working with people with lived experience and you represent a profession that has denied them liberty? How are you going to communicate with them? Or if, they don't use the scientific terms, how are you going to make sure you don't just rely on the safe, reliable, convenient voices who speak the language of science and, diversify and reach out to people who are not included in research at all? It's all a work in progress, I suppose, but something that I enjoy working on. And I'm wondering, as I come towards the end, who is this for, really? I think it's for the benefit of patients, people like me, and what is co-production about? Well, it's with people with lived experience. I think it's so much more than that. My vision for co-production is that it's about cross-disciplinary working and moving from sort of multidisciplinary, where disciplines are side by side. I've had the psychiatrist say this, the gastroenterologist say that, nobody really putting it all together. Or I've been in research meetings where even people working on animal studies on the same mental health condition sitting next to each other actually don't know what each other's work are because they're so hyper-specialised. Don't they need co-production as well? So who is this about? I think it's a sort of shift in the field that we need towards that truly transdisciplinary, interdisciplinary, where we go and create something else that brings together all of the multiple perspectives rather than somehow expecting them to be alongside one another and integrate that way. And that involves getting out of our boxes, getting out of our ways of seeing things that are convenient. Because I think the future of mental health science is going to be in those spaces in between. It's not going to be in one lab. It's not going to be, finding the right variable or the right measure or the right treatment, because human experience is really complex. It's contradictory. So I think we do science and the public actually a disservice if we try and erase that complexity in the name of sort of tidy science, yeah? And even if we do take the philosophical view that there are natural kinds of illness and we just need better instruments and we'll be able to neatly define everything, these are all things that are experienced and interpreted by unique people. We can't erase that subjectivity either. So I think that the future of mental health science really is in thinking about growing the methods rather than that reductionist approach, to think about how can we actually get inside that complexity, that uncertainty, and not avoid it, not be desperate to know, which is something that we want, and desperate to help, which is something that we all want, but also to admit where we currently are, because I think it's in that unknowing that the exciting stuff really can happen. So yeah, that's it from me really, and I hope to understand the remaining talks. But I think in order to bring it out of the lab to meet people in their real lives, we have to bring people together. Because I can give one example that's relevant to animal studies. Recently, I was involved with a project, and I was like, that measure of anhedonia, that's not what I think it is at all in my experience. Or listening to you, I'm sort of like, what about later stressors? Not just this intense focus on early childhood, right? These sort of aha moments that can come about from the phenomenology, and it's not at all unscientific or anecdotal to really tap in and tune into that phenomenology of people's experience. So if there's any time for any questions, I don't know. Yeah, thank you.
Thanks very much, James. I like your plea for untidy science. We've got a bit of time for questions.
I guess this isn't really a question for you, James. I guess it's more an observation that this idea of the complexity of human existence is something that impacts on lots of different health conditions. And thinking of animal models of this, we are obviously constrained by what the Home Office essentially will allow us to do. But thinking, trying to model those things in terms of things like binge drinking on a Friday and all these kind of things, how would you do that in an animal model? And I guess the question, it's not really a question, it's an observation that we need to maybe work with the kind of regulatory systems to think of a mechanism for getting around some of the constraints that are put on us in terms of modelling, the reductionist approach to using animals, the specific requirements for living conditions and stuff like that. And I guess, is this something that the Wellcome and other funders might be willing to help advocate for? I guess that's what I'm asking.
Yeah. I think that they are. I suppose that even the most minute finding, just having somebody with lived experience in the room, you might think, oh, they won't understand the science. Well, there's training that can be provided. You know, people can find a mutual language. But having people in the room who are coming very much from the ecology of this, what is it like in context? What is this brain function like in somebody's body, in society, in the world? Like that perspective being in the room, being present, I think that's where people with lived experience can add value if it's done well. It's not always done well. And yeah, I think otherwise we get so far down that hyper-specialised burrow, don't we? And forget that, oh, this has to relate to context in which is probably changed by the time that you've published the thing anyway, and regulation, all this kind of stuff. I know you'll probably hear more from Welcome later on about how to address this.
Yeah, I don't know, Niall, if you want to add anything on this from the Welcome perspective, given.
I don't know if everyone can hear me. I'd say that if you come to the plenary tomorrow, which I think is around mid-morning, you will hear much more about this, and we will discuss it further. But yes, I think that it's important to join everything up as much as possible from lived experience, and not just as James has said. I think that it's important that with lived experience we move beyond the idea about simply people's stories, and also think about the technical challenges which that can bring, and give people the appropriate training to make that challenge. And then, as the questioners just asked, thinking about the implications for that in terms of things like regulation and laboratory practices and so on, and trying to find all the links in that chain to really get to the best models and produce the treatments that we need.
And it's just one comment is that I started saying, let me tell you a story kind of thing. I never do that in my lived experience work. I'm not very interested in telling my story unless it's like something helpful for the media. I think it's about how that lens of subjectivity coming into the room as part of the sort of methodological picture, and thinking how is subjective experience applied to a specific task and to some specific research question. I think that's something we need to think about developing in this field of lived experience, so that it's not just tokenistic or like, oh, that's nice that you told us a story. Like, was that really my story? No, it was kind of saying, look, there are big epistemological questions going on here, and that applied approach to lived experience rather than extractive.
Any more questions?
I take it as a good sign, like stunned silence or something.
I might just add one. One question is, aspects of this work are quite novel, bringing lived experience into the sort of most basic aspects of mental health science. Have you generally had a positive reception? Is there skepticism, or do people generally accept that there are limitations and that there's a role for lived experience to play?
Yeah, so only in the last year have I done stuff that's really sort of very highly scientific, more so, like triangulation meetings, this kind of stuff. And I think generally people have been like quite surprised that people with lived experience can engage with it. We've had training provided to us, so looking at how bias is assessed and all this kind of stuff, and what does, how do we interpret this data from our lens? But I think people have maybe had moments where they thought, I assumed, or have said, or admitted, I assumed that people with lived experience wouldn't be able to be involved. Now, I am somebody who's like naturally interested in this and educated and able to be articulate at times, but maybe not after lunch. But I think this is about really sort of building a diversity of voices as well, because we found some really promising things like, oh, there is real value here. Sometimes it's been sort of like the glue of meetings that we've had, but it's then how does this workforce develop? That's something Welcome, I think, is at the front of, and how, what skills do they need, what support do people like me need, even if we appear competent, etc. I don't know why I'm looking at you, it's because you asked a question, but I'm addressing it all to you. So yeah, I've had a really positive experience, and I feel like it's really refreshing actually, compared to a lot of other work that I do more in clinical spaces, where it feels like you confront the injustices that might be present in resource-strapped services at the moment, and it can feel really negative. Like, fighting the negative is really exhausting. There is an emotional burden to doing this kind of work, but creating the new is completely different. It has a different feeling to it. It's an exciting thing, and I'm sure we're all here today because we're excited about the future of mental health science.
Thanks, James. I think that's a really positive way to end. So let's thank James and welcome Rachel Hill.
Thank you.
Thanks, Kathy and Niall, for inviting me to present today on collecting and evaluating animal models in mental health science and building an online resource for researchers. This is a welcome initiative and it's led by myself and Professor Suresh Sundaram from Monash University in Australia. And it's also a collaborative with the Consortium for Preclinical Psychiatric Research, as well as Professor Falk Scholar from RMIT University in Australia and ThoughtWorks. And we have no conflicts of interest to declare. So mental disorders are one of the top 10 leading causes of global burden of disease in 2019. And this graph here shows that mental disorders affect people of all ages. They also affect people of all ethnicities and all socio-demographic backgrounds. And importantly, there's been no change in global reduction of burden since 1990. And this is largely due to the fact that treatments remain mainly symptom-based and do not actually modify the course of the disorder. They often come with substantial off-target side effects, which make compliance rates low. So it's clear that there is an urgent need for treatments that actually modify the course of the disorder. And there's been recommendations from many sources that a research focus on causal pathways in mental disorders is needed to really reduce global burden. But how can we identify causal pathways in disorders of the brain whereby we can't actually see into the brain when people are really most at their sickest? There's a number of different techniques that we use. Human brain imaging can help to identify specific regions that are impacted, and using PET imaging, specific markers or neurotransmitters that are altered in disease versus healthy participants. And this is an example of dopamine uptake in a patient with schizophrenia versus healthy controls. And a more recent example of M1 binding in the dorsolateral prefrontal cortex. But of course, we are limited somewhat in brain imaging in our ability to understand really complex biological pathways. Post-mortem studies can help to identify key cells and molecules that are altered in specific diagnostic groups. And certainly with significant advances in single nuclei and spatial sequencing, we're really able to see these molecular changes at a much higher resolution than ever before. However, we do still need to consider with post-mortem studies that they generally represent the end stage of a disorder. So how do we really know if these molecular changes are causative or circumstantial? Stem cell or organoid models derived from patients can help to understand how disease-related genetic backgrounds can impact specific cellular and molecular pathways, particularly during early development. And this is an example of ventricular progenitors that are disrupted in schizophrenia patient-derived organoids. However, an obvious limitation here is that we cannot assess behavioural outcomes, which is the main way that we diagnose these disorders. Animal models are used to investigate the role of different genetic and environmental risk factors on brain development, function and behaviour. Here we can assess the biology of the brain at multiple time points. We're not restricted just to the end point, and we can link with behaviour. We can also use animal models to test new drugs. However, at the end of the day, they're animals and they're not humans. So all of these techniques or approaches are highly useful in trying to identify causal pathways. But what is really needed is for all of these approaches to be working together to really try to piece the different pieces of the puzzle together. So with this in mind, in 2024, I established the Consortium for Preclinical Psychiatric Research. This consortium now consists of over 50 researchers from across Australia, representing over 14 different universities and institutes. And this is just a few of them here. It includes researchers in imaging, post-mortem human studies, animal model studies, and stem cell studies, with all of us looking at psychiatric research. Our vision is to improve biological understanding of mental illness and fast-track the discovery of new treatments through collaboration. And one of the first goals that we set as this consortium was to establish a national knowledge bank of imaging, post-mortem samples and preclinical models. So this knowledge bank contains tabulated information on all of the post-mortem samples currently available in Australia, imaging data, as well as preclinical models. And we also have a tissue or banked tissue information as well. And if you're interested in having a look at this online resource, the DOI is listed there. So something that we noticed when looking through our knowledge bank was that we had over 55 animal models relevant to psychiatric disorders. And this is just in our tiny little corner of the globe in Australia. So when we consider how many animal models must there be out there globally, there must be hundreds that are relevant to psychiatric research. Each animal model is modelling different genetic or environmental risk factors or combined hits, and really providing very important information on causal pathways or potential causal pathways in mental disorders. But it does tend to leave researchers wondering which animal model is the most appropriate model for their specific research question. Similarly, multiple assays tend to be used to assess behavioural domains, and these are just a few assays that we use in my lab. So which assay should be applied to assess the validity of the model or to test new drugs? And how can we consolidate our understanding of causal pathways for specific behavioural domains if the way in which we assess these domains differs across laboratories? So this is where the Wellcome initiative happened to come across our path last year on collecting and evaluating animal models in mental health science and build an online library of animal models resource for researchers. Here, lived experience is really central to this initiative and will guide which symptom domains are most relevant to patients experiencing the conditions being modelled, and therefore guide model assessment and prioritisation. So the first phase of this initiative is to evaluate and rank all the models and assays for the domains of depression, anxiety and psychoses. So these are symptom domains of depression, anxiety and psychoses that of course can be present across multiple diagnoses. Phase 2 of this initiative is to build a world-first interactive web-based platform with AI capabilities that will enable a wide range of users to easily identify the most useful model and relevant model for their specific question and the most relevant assays to use for their specific research question. And again, lived experience is really important here to guide the user-friendly platform development. So we're currently in phase 1 of information gathering and evaluation of current models and assays. So we've formed 3 major working groups for the domains of depression, anxiety and psychoses, and these include both preclinical and clinical researchers. We also have established a lived experience panel that will be guiding each of these working groups. Now, these working groups currently consist of Australian researchers, but we really would like to invite international experts to join these working groups. So if you're interested and you feel like you can provide your expertise to any of these working groups, please do contact me. So extracted data for assays for depression, anxiety and psychoses will be structured through a scoring matrix such as this example that I've provided here. So this is an example of behavioural assays related to anxiety. I've just provided a couple of assays as an example. And we're going to be providing a scoring matrix such as this one here, looking at things like relevance of symptom domain to the disorder. Obviously with this one, lived experience is going to be really important in guiding us as to, is this elevated plus maze, does it have any relevance to what you're actually experiencing? Such as what James was mentioning about the anhedonia tests previously. We're also looking at things like predictive validity, publications using the assay, citations, average effect sizes, feasibility or accessibility of the assay. Is it easy to establish? Is it widely used? And also a major one is ethical considerations. So obviously following the principles of the 3 Rs, replacement, reduction and refinement. So if it requires a reasonable number of animals and is not distressful to the animal, then it scores much higher, obviously. To rank the most useful models, we will follow a flowchart such as this one described here, which again includes an ethical evaluation, a scientific evaluation, so reliability and replicability, models with face validity, so similarity with what is modelled, and models with predictive validity. Sometimes predictive validity can mean whether it responds to current treatments, and obviously this depends on the model. If you're looking to model, for example, treatment-resistant schizophrenia, you probably don't want your model to respond to current antipsychotics. So there is a degree of flexibility required here, and this is why we have our expert panels involved here of both clinicians, preclinical researchers, but also our lived experience panel to guide us here. Does your model show construct validity, so sound theoretical rationale, and model with external validity, so generalisability across labs and environments? So importantly, all of the models will be run through this flow chart in a systematic manner. So the second phase of this initiative will be to develop this library of models platform that we're hoping that researchers and lay people and the community and as well as industry can use to really go onto the website and ask it questions and ask it, which is the right animal model for me, which were the right assays to use. He will be using centred design principles to create a web portal that is really highly intuitive. So it's a real focus on accessibility and inclusion. And the portal will support information access and retrieval for users with different levels of ability, so from really high expert researchers through to lay people. It will use AI-enhanced retrieval augmented generation to resolve natural language expressions of information needs. So this means it'll take sort of information from outside sources and make sure it's the most up-to-date AI that's being generated and answering the questions. And filtering will be based on specific assay factors as specified by our expert research groups. So I'm a behavioural neuroscientist by trade, and I've never developed a website before. So what we did was we partnered with this wonderful company called ThoughtWorks, who have developed websites in collaboration with Amazon and some of the big banks. They also happen to have done a lot of work with academic partners, which is why we thought they would be a really good fit here. And this is one such example of a collaboration they did using digital tools to reduce the global threat of mosquito-borne diseases. So they actually developed an app that's currently being used across 9 different countries, which supports field activities such as releasing and collecting and monitoring mosquitoes. So they'll be working with us to develop this platform. So we're really hoping that we're, at the end of this initiative, we'll develop an interactive platform from which researchers globally can precisely formulate their question, first of all, and identify the optimal model and assay to answer their question. A platform that really showcases the value of preclinical models and extends their usage and understanding to a broader international research community and really helps people to collaborate across the world. It'll be a dynamic interface, we're hoping, where new models can actually be added. So it's not just a website that we develop and it's just got all the current models. We want to be able to add models to it and for it to be constantly updated and integrated with new data analytics. To really facilitate a truly global collaborative network, provide guidance on the most useful models for testing new treatments for depression, anxiety and psychoses, and be widely accessible to public and private sectors, to fast-track translation from preclinical to clinical testing and provide new and improved treatments for people with a mental illness. So how can you be a part of this? If you're interested in being part of the expert working groups, we meet every 2 months for approximately 2 hours, and you would be compensated for your time, so please do contact me if you're interested. We are also currently hiring 3 postdocs for this initiative. If you fancy a move to Australia, please contact me. And finally, I just want to finish by running you through another initiative that's recently been funded for the Consortium for Preclinical Psychiatric Research, which is applying an omics approach to interrogate our large sample banks. So our sample banks currently contain blood, brain and patient-derived stem cell samples from our banked patient samples, as well as brain tissue from our animal models. Importantly, all of our sample sources have really robust diagnostic information, and all of our animal models have robust behavioural phenotypic information. So with all of our banked samples, we've just received funding to apply a harmonised omics approach, which will include methylomics, transcriptomics and proteomics, to hopefully identify new causal pathways, potential brain mechanisms linked to specific symptom domains, identify new targets potentially for therapeutic development, and identify biomarkers. This first phase is specifically for schizophrenia, so for schizophrenia subtypes. This is sort of an iterative approach where newly identified causative pathways can then be modelled back in our stem cell and animal models, and newly identified drug targets can also be tested in our animal models. So this is currently an Australian initiative, but we do have hopes that we could potentially make this a global platform of omics data in preclinical models that will really help in sort of finding causal pathways in mental disorders. And I'll leave it on that. Thank you.
Thanks, Rachel, for discussing these important initiatives. We have a couple of minutes for questions.
Hi, thanks, Rachel. It was a really interesting talk and initiative. Can I ask you what your thoughts are on methodological differences? I think there's probably no lab that runs their tests the same way as any other lab, and we have huge variability. Maybe we're not the worst, but in behavioural neuroscience, I think, there's a huge amount of variation, even just how the equipment is designed. So how will you kind of manage that, I guess, when you're rating?
Yeah, it's a really good question on how we manage that. And it might even be that what it leads to is certain guidelines to be released on how people report, first of all, their behavioural tests and how they're run and the environmental conditions. It's something that our working groups are going to have to work through systematically with each assay, even within very specific assays. For example, a Y-maze test, people have very different intertrial intervals, and so yeah, it's just something we're going to have to work through systematically with each singular assay.
Hi, thanks very much. That was super interesting. The bit that I'm wondering about, if you could expand a little bit more, is so you talk about creating this platform that enables you to sort of come up with like the optimal assay for a particular phenotype, say. How do you avoid that becoming incredibly subjective? Because the perfect assay for a phenotype may not exist, and two, how do you avoid it becoming certain tasks? The one that comes to mind is like the forced swim test, right? Which is very heavily cited. Lots of people have used it, would argue, but that's the best one. How do you become like very sort of historical type assays become very entrenched?
Yeah, it's a great question. And I foresee that it's not likely that we're going to end up with one golden assay or one golden model for each domain, that it's more likely, what I'm hoping is that we're going to advise on specific combinations of assays that might be useful. So, 3 or 4 assays that would be helpful for anxiety or anhedonia. And I'm not even ruling out, it can't just be restricted to behaviour. It could be that, combining with an electrophysiological assay could be really beneficial. And I think that by incorporating, these lived experience panels as well, I think this is where it sort of differs from previous maybe attempts to do this in single labs or single groups, where we're actually getting information from people living with these experiences that can tell us, actually, it's this aspect that's important, so let's focus on that.
Thanks. It's really inspiring, exciting to hear about this joined up working. Picking up on, I think, a similar theme that was discussed in the previous talk. I'm just wondering about this concept that the assays need to be non-distressing for animals. I'm not an animal researcher, because depression, anxiety and psychosis are deeply distressing for patients, but also for their friends and family, sometimes more so, particularly in acute psychosis, than they are for patients themselves, maybe akin to animal researcher kind of relationships. So I'm wondering if that's a fundamental flaw or limitation, and how you tackle it.
Yeah, I mean, it's always going to be a limitation, particularly within sort of animal models of depression, you're looking at things like behavioural despair, which how do you create behavioural despair in an animal? It's never going to be nice. And it's something, I know in Australia, for example, the forced swim test is banned. We're not rightly so allowed to do it because it's not a good task. But when you talk to people with lived experience, they're sort of like, well, what does that have to do with behavioural despair? It just seems like an odd assay anyway. And so I think, I'm hoping that out of this we actually might get different approaches to some of our assays. These are assays that have been entrenched in behavioural neuroscience for 20, 30 years, some of them. And perhaps with these different perspectives, we actually might start to develop novel ways of assessing things like behavioural despair and anhedonia.
I think we'll have to move on to the final talk, but please save those questions and ask them in the panel discussion later. Thank you very much, Rachel.
Thank you.
So now we have Dr. Georgia Salanti from the University of Bern, who is joining us online. So I'm hoping she's there and will be able to share her slides with us. Is there a problem with the sound? Yep, Georgia, just hang on. We're going just trying to sort the sound. I wonder if, while we're sorting this, Rachel, would you mind, because there are a couple more questions. Sorry, worst nightmare, more questions. I think so that we use the time.
Thanks, great talk. So coming into behavioural neuroscience, I was originally an electrophysiologist and then trying to combine and getting into it sort of thing. But what I've noticed is no matter what you do, when you get reviewers, there's already one reviewer that says you should be doing an extra 4 tests. Yeah. And I sort of think, I guess what we need is a directive. I guess and maybe this might be like, this is the minimum, and then you can cite that sort of thing if it was accepted globally. But also the other side of it, it may say that you need to do these 4 tests, but in smaller labs with limited funding and limited, they may not be able to do that as well, or have access to collaborators. So I'm just wondering whether it also might be another way of connecting people.
Yeah, and that's what we're hoping is that it's, as we sort of start to add models to the platform, that we can reach out to the authors of the models and sort of say, would you be happy to be contacted, attach contact details to the models, to the assays that are developed, and hopefully connect people. And hopefully, ethically, that'll actually help in reducing the amount of animals that are used as well. If there's a couple of models that are really good, and one lab has all of the behavioural equipment, let's all do everything in that lab and not replicate these animals, in 5 different labs and hundreds more mice that have to be used. So we're hoping that it'll do both of those things, connect people and also reduce the amount of animals that we use.
AV, do we have Georgia yet? Not yet, okay. I think we'll give Rachel one more question, if that's okay. And then if we still can't get Georgia, we might flip the order and do a panel discussion first. Sure. Okay.
Hi. I have a question about the platform itself. I was wondering if you plural you as a group, we're intending for it to be like open source code or something so that other people could repeat what you're doing with, say, like models of chronic pain or something.
Yeah, absolutely. I think we'd like for that to happen. Niall's nodding his head, so yes, we'd like for it to be open source. If this can be replicated in other disorders, fantastic. If it can be used more broadly, even better.
Thanks, Rachel. So where are we? Do we have Georgia yet? So I think maybe it's so that we use the time. Sorry for the change of schedule. Let's have a panel Q&A with the three that are here, and then hopefully we'll be able to get Georgia online in the next sort of 10, 15 minutes, and she can give her talk. Okay, so please welcome Jeff, Rachel, and James to the stage. So thanks so much for your fascinating talks. I wonder if we have any questions for the panel from the audience. Straight in there.
So I've been interested to hear, as something of an outsider in this area, that the word reductionist is seen almost as a dirty word, something you really want to avoid. I accept that real biology, real medicine is extremely complicated, but if one looks at the history of science, reductionism has delivered almost all of the big discoveries that have been made, and that includes in neuroscience. So while I accept that you may be studying a microcosm that has very little relevance to the higher level, it seems to me that reductionism cannot be left out totally. Even if one brings in things which sound important, like lived experience, one has to be able to isolate phenomena in order to be able to study them meaningfully. In particular, one has to stop oneself by being misled, so by being able to ask a question that will tell you you've got the wrong answer there. So, falsification is an important thing, and celebrating complexity and studying it at a high level quite often means you solve nothing, but you get lots and lots of data. So that's a kind of devil's advocate sort of way of putting it.
Yeah. Can I respond to that? I agree. I think the strength of animal research is that it's mechanism-based, that we absorb ourselves, we focus on the mechanism. That means it's a highly reductive process. We've heard this word model today, a term I don't necessarily agree with. Modelling what exactly? We've talked about the complexity. This is impossible to get at in an animal, frankly. So what we should be doing is saying we have a mammalian nervous system, and to understand it through a reductionist approach is a mechanism-based approach. But that's my take on that question. It's a very good question.
Yeah, I would agree. I think that I might have used it, the word reductionist, in maybe slightly perceived in a negative way. I think it's always about both and the work of somebody like me talking to a neuroscientist is dialectical, isn't it? We're coming from very different perspectives, and I think I'm calling for a sort of both and, like, let's maybe rebalance and bring people into the same space. And there was so much, in one of my main frustrations in eating disorders, which is the area I experience and work in the most, is that there is so much primary biology that we don't know in these conditions, and actually many people, I think, clinically who aren't that interested in the biology. So I'm always like, look, let's find out more, and finding out one very specific gene mutation that I have, being diagnosed with a rare genetic condition, actually helped me to put together those pieces of how do the symptoms that I experience. it was a really important part of understanding the whole picture. So I feel like it has to be both, and somebody saying sort of, hang on, what about us and the lived experience is not to say like, you know, the reductionist stuff is bad. It's just also this, and we need to keep adding that sort of expansionist, which you know, if that makes sense. Yeah.
Rachel?
Yeah, no, I think it's the reason why we have ended up with hundreds and hundreds of models. We don't have hundreds of models of depression, of anxiety. We have hundreds of models that are modelling genetic risk factors of depression, anxiety, or psychosis, or environmental risk factors, and every single one of those models is telling us something really important about one of the pathways that's involved. So it's certainly, you know, I totally agree. We do.
I cannot hear you because your.
Is this not on?
It sounds like we might have Georgia. Georgia, if you're there, do you have any points to add to this important question?
Can you hear me?
Yes.
That's fantastic. I can hear myself as well. So am I allowed to leave? Oh.
Hi there.
I see myself in multiple screens now. I'm not sure what to do. I need to mute one of the two. Probably.
Maybe if you can mute. Yeah, sorry, Georgia, I think.
Yeah, mute is not what is needed. I think I need to decrease the sound or kill the sound from the Zoom or leave Zoom altogether, but I'm not sure that's a good idea. Can I try to leave Zoom and stay only in... Okay,
Yeah.
already I'm kicked out of Zoom. Fine, but still it works. So you see me all fine?
Yes.
Good.
Yeah. Just wondered if you had any thoughts on that last question about challenging the idea that reductionism is necessarily a bad thing, and saying how important it is for scientific progress.
I have to confess, because I've been trying very hard to make things work, I haven't thought about that.
That's completely fine. Okay, let's move on to the next question, and hopefully you can input there.
Freak, and that's why I became an experimental scientist. So it's hard for me to say this, but I think we've controlled the biology out of our models in some respects and the tasks. And we do need to compromise, I think. So I'm not against reductionism, but we don't consider individual variability enough. What are our outliers are actually probably some of the most interesting cases. When we look at things like, from Jeff's really interesting talk about maternal separation, you see different responses in the offspring. They don't always show resilience. Some will show resilience, some susceptibility, and we don't always capture that, and we need to do bigger group sizes, which is not great for NC3Rs. But I think we need to think not just about looking at group comparisons, but I think by reductionism, we sometimes go down the wrong route because we don't include those outliers, and we'll make an opinion and say, okay, this is the mean is showing us this, when actually it probably wouldn't be if we did 20 more animals, for example. So I think we have to find some kind of balance between that and embrace some complexity in the model and not just go always for a simple approach. But it'd be interesting to hear what the panel think about that.
Thank you. Yes, how do we model individual variability?
I mean, I can't speak to the animal studies so much, but certainly clinically, I think the outliers are perhaps where some of the answers are as to why things aren't working in the mainstream. And we have services and treatments that are designed for these ideal patients that aren't the ones that really walk through the door. So, like in my case, thinking of like my hypermobile condition affects my stomach motility, which means all kinds of things gastroenterological, and that is anecdotal. It is specific to me. But then I've gone on in other parts of my work into inpatient clinics and just asked people like whether they're hypermobile, and half of the inpatients were sort of like not thinking about this in their treatment and what impact that might have. And like I'm sort of thinking, well, that's an area where then there could be some really specific research questions there that are reductionist in a really helpful way, mechanisms, etc. But I think the sidelining of the things that don't fit, like there's a lot of missed opportunities there, and that's where the complexity and the unknown and the exciting stuff might be. Don't know how it works with the animals.
I mean, I'll just comment that there are a number of people in this room who've made their careers from looking at individual differences. A dimensional analysis is a powerful analysis. The way I see this working too is we have all of these paradigms and procedures we use in rodents. We're not always clear, and Rachel was making this point, what they're telling us, what the phenotype is. But if you have the same animal doing lots of different things, what you do is you create variance. And then you can have a higher order clustering algorithm that can look at common biological substrates and mechanisms. So you have to do a bit of both. You have to look at individual differences, but you can't always look inwardly at a single animal. You have to draw lessons from a cluster if you can. So, and that will translate. And I hope you've seen some examples of that today. That's my take on that question, Rachel.
Yeah, I don't, if this is this one working, I agree. And you know, the amount of times I've looked at social interaction data, for example, and there's that one mouse that just sticks with the other mouse the whole time. You're like, what are you doing? You know, and you want to know what's going on with that mouse. I look at infection during pregnancy as a risk factor as well, and you have some mice that almost look like they're actually resilient. We had a whole cohort that actually had improved spatial memory after infection during pregnancy. You know, so looking at things that are not quite fitting with your hypothesis or outliers is so important. Unfortunately, we do need more numbers. I know there's a large group in Australia where they do large numbers of mice and look at the ones that are resilient compared to non-resilient. It would be great if we can look at them more, but yeah, it requires more numbers, which it's that fine line between keeping it ethical and also exploring.
If I can chip in, to say that from my perspective, I'm looking more at groups of animals, that is studies, and you can look at the microcosmos of a cluster of animals and observations, and then explore the individual differences and talk about outliers. Or you can have also a complementary bird-eyes view where you look at the within study variability, but also the across studies variability. And you can talk about the individual effect, or you can talk also about the mean effect of, let's say, an intervention or whatever it is that you study. And from our perspective in Galenos, where we are working a lot with meta-analysis, we are considering both. And sometimes it is very useful to put the individual variability into context if you consider how this variability manifests across multiple similar studies.
Thanks. I think we'll have to leave the discussion there so that we can give Georgia enough time for her talk. Thank you very much to the panel. Okay, Georgia, whenever you're ready.
I am ready. I wonder whether you see my screen too. I have shared my screen. Yes, good. So you see the title, A Framework to Assist Research Prioritization, right? Yes. Correct? You see the title slide? Yes. Yes, okay, thank you. So I move on. I'm going to talk to you about the project Galenos that was inspired by the realization that funders of my biomedical research, but also researchers themselves, disregard the existing evidence when they set their priorities, when they choose how much money are going to give to which research idea, or what grant application they're going to write for, which idea they are running after. And that leads to research waste or minimal added value of new research. And so the Wellcome Trust is funding a project called Galenos, that stands for Global Alliance for Living Evidence in Anxiety, Depression, and Psychosis. You can Google it and see our website, where we are trying to identify promising routes of new treatments for mental health problems or novel diagnostic tools and make more accurate predictions. Not necessarily to inform directly the clinical practice, but in order to inform research prioritization, to say which is the future or which are the new treatments that we should prioritize in the pipeline, for example. And our main methodological vehicle is a new type of living evidence resource of early phase studies. So, although many treatments exist for several mental health conditions, and in particular for depression, anxiety, and psychosis, that is the focus of Galenos. So, although many treatments do exist, some other conditions, they are orphaned, or new treatment options are clearly needed. And I will take you through the example of TAR1 agonists, that they have been thought to be promising alternative to the existing antipsychotics because they were shown to have, or they were assumed to have fewer side effects than the existing pharmacological treatments. However, we found only 4 early-stage small clinical trials with data in humans, and we identified them using a systematic review. Most of you, I guess, know what is a systematic review. We are using, however, a living systematic review. This is a technique that, unlike the traditional systematic reviews, that they are static, the living systematic reviews, they are dynamic and they are regularly updated to include new research funding. And we have created template protocols that they are followed in all our living systematic reviews. You can look at those templates online. So, and this is our framework in order to answer the question, is TAR1 agonist a promising research route for treating people with psychosis? So first we will do a synthesis of human studies, then we are doing a synthesis of animal studies, then we triangulate the evidence, and then finally we update the evidence. So in the synthesis of human studies, as I said, we follow standard approaches of living systematic reviews. And for this example of TAR1 agonists, we identify, as I said, only 4 studies that they are in psychosis, some others that were in healthy volunteers or in Parkinson diseases. And they are, all 4 of them, they are rather small and of limited duration. And these studies, they are, however, of low risk of bias, as shown by the green traffic light that is next to these 4 studies by Coblan and another 3 that they are NCT, they start with NCT. So, and this is what the synthesis of these 4 studies shows. These 4 studies, 3 of them use ulotaront and 1 use another drug, but if we put them together and ignoring the little differences between the substances, we see that the common effect model suggests that the standardized mean difference of improvement of schizophrenia symptoms is larger with TAR1 agonist than placebo, although there is quite some heterogeneity as reflected by the rather wide confidence intervals in the random effects synthesis. Another method to look at the association and whether actually these the TAR1 agonists are associated with decrease in overall symptoms is to look at the dose-response effect. But given the few data that we had, it was not really possible to draw a conclusion. Overall, we will say there is a very mild association of increase, but not really much. However, why should we restrict ourselves only to the human studies when there are more than 125 published experiments in rodents? And we collected them all. We looked for studies where the animal model was done either by a genetic intervention or using psychostimulants, and then the TAR1 agonists were given to the animals. Maybe there are arms additional to that, like antipsychotics or placebo, and then we monitor how some outcomes change, like the increased locomotor activity, etc. We search for studies like that, and we extract the data, and we synthesize these animal studies. And you see here on the screen, or you don't really see here on the screen, because there are so many, 125, as I said, experiments that compare TAR1 agonists versus placebo in models for psychosis. And what you see is the impact on locomotor activity. And you see that there is a clear advantage of the TAR1 agonists. However, these are data on animals. These are data on locomotor activity and not on psychotic features, and that's why we need to put them into context together with the human studies. Here I just present again the results from the animal studies grouped by the compound of TAR1 agonist, and that's again the total effect. There was a lot of heterogeneity as reflected by the prediction interval, which was quite wide and included the standardized mean difference value of 0, which means that we cannot really very, very confident reject the null hypothesis. But given the amount of data and the confidence interval, ignoring the heterogeneity, our conclusion was that the animal data favors TAR1 agonists, but with a few limitations. The most important of which is that we detected potential bias due to missing evidence. That means that there was publication bias. Experiments that did not show an advantage of TAR1 agonists were probably suppressed and not identify them. However, the data from the animals saw a very clear dose effect for locomotor activity, and they show that 1 unit of increase in standardized dose was associated with an average increase in the effect size, so that's the standardized mean difference, by 0.20, and that was also statistically significant. The second very important downside of the evidence from animal studies is the high risk of bias. We evaluated the studies according to 10 different domains that you see here on the right-hand side, like allocation, sequence, generation, the baseline, similarity, etc. And in most cases, we could not really evaluate it because it was not reported. So reporting quality and most probably conduct quality of the studies was rather. So, and then we did a triangulation of the evidence. And here I need to make maybe a pause to explain what triangulation of evidence is in epidemiology. Before going to epidemiology, I will start with the first time that the term triangulation was used. And also telling you that Galenos is not just a name, it was a real person. Galenos was a Roman Greek doctor in the 2nd century AC, who was very much interested on psychological conditions. And in 200 BC, Eratosthenes, another Greek but mathematician this time, measured the circumference of the Earth using properties of triangles. So actually what he did, he placed some sticks in different places in what is nowadays Egypt, and he measured the shades and the triangles that they say create with respect to the position of the sun, and he measured very accurately the circumference of the Earth. And that is associated with a more general concept of triangulation that we use in this project. So triangulation refers to the methodology that when you want to measure something, but the measurement is very difficult or impossible, then you can measure other things that they are easier to measure, like sticks and shadows, as Eratosthenes did, or biased, non-causal effects of TAR1 agonists in animal studies or in early phase studies, and then combine these suboptimal measured quantities in order to infer about your main target, so the true effect of TAR1 agonists. So triangulation has been used quite a lot in social sciences and in psychology, where it involves qualitative and quantitative evidence. There are very few examples in epidemiology, but in Galenos, we have the first one that we use human and animal studies from systematic reviews. We consider that these two types of evidence, they are independent and they create two different evidence theses. We consider the results together in terms of direction and strength of association, but central to triangulation is the evaluation of bias within every evidence thesis and its possible direction at the systematic review level, not necessarily at every study separately. So we have developed a triangulation framework where bias can occur due to study limitations, that's the risk of bias in the included studies, the reporting bias, so simply put the publication bias, and indirectness, which refers to whether the population study, the drugs, the outcomes are not exactly as we want them. And you can already imagine that indirectness is a big problem in animal studies because the populations, they are not human. So, and that's the backbone of our triangulation framework, which is we call the summary of the evidence. For any particular outcome, we summarize the evidence from the human studies and then from the animal studies. And we match the outcomes as best as we can with respect to their similarity. So, for example, the overall symptoms could be approximated, even if it is a bad approximation, that can be approximated by the locomotor activity of animals. And then you see the strength of the association as measured by the standardized mean difference and the confidence interval for the human studies and the animal studies. Then we discuss about the heterogeneity. So human studies did not really have much heterogeneity, but there was quite some heterogeneity in the animal studies, and there was some dose-response relationship in animal studies, but uncertain in human studies. And then the three main components of bias, the within-study bias, we discussed that with this traffic lights graph that the human studies have overall low risk of bias, but the animal studies have moderate risk of bias, and they are likely to exaggerate the effect of TAR1 agonists. And exaggeration in the animal studies can occur also because of a particular across studies biases, that's publication bias, and indirectness, obviously low risk of indirectness in human studies, but moderate risk for animal studies. And in order to evaluate the risk of indirectness in animal studies, we have developed also another framework or tool that considers factors relating to animal population, model induction, intervention, outcomes, and considerations for homological, ontopathogenic, etc., validity of the studies that we include. And there is also room for some other types of biases, like,
Georgia,
for example,
just 2-minute warning.
Yeah, I'm nearly done. Some other bias that you might want to consider. And then we organize a triangulation meeting where people with lived experience, expert in animal studies, clinicians, and methodologists meet. In that case, we met in Wellcome Trust in London in order to discuss all this evidence and decide whether this particular research path, TAR1 agonist, is a promising one, and we should invest in more studies, more experimentation with that. And finally, we update the evidence. In our protocol already, we do have a rather complicated flow chart to help us decide a priori and avoid judgment biases, when and how and under which conditions we will update the evidence. So, just to wrap up, the funders of biomedical research very often disregard the existing evidence when setting their priorities, and that could lead to research waste. And evidence from research, from systematic reviews of animal studies, is often considered in isolation, and so does the research from human studies. But we suggest that the best way forward is actually a living systematic review with a triangulation process to put together evidence from humans and from animal studies in order to answer questions about emerging technologies in a timely and transparent manner. And I think I finished even without using the full 2 minutes.
Thank you very much, Georgia, and thanks for bearing with us with the technical issues. I think we've got a couple of minutes for questions. At the back.
Thank you. Thanks very much. It was really interesting and a really nice, I thought, kind of wrap up after the question about reductionism. And actually, you know, the best idea is to combine a whole load of different types of data and work out where there is matching evidence. I was just curious how you're using the tool, how you're deciding, you know, what targets to analyze using this system.
All right, so how we set the research question, so how to decide which project to do. Yeah?
Yes.
Yes, okay, sorry, I cannot always hear. Yeah, so we have a panel consisting of clinicians, people with lived experience, and experts in animal studies, and priority is given to the opinion of the people with lived experience, where they come up with various ideas, and then they sit together and prioritize with the clinical and lab experts, you know, with respect to feasibility and timeliness and relevance to the people, but also researchers. And I think, I don't know whether Niall is in the audience, but I think we also had at least one, like a top-down request from Wellcome Trust to do a particular systematic review. But all other reviews, they are done in this manner of co-production.
Yeah, I can fill in on that. Yes, there's one particular living systematic review which is on internet use, so the quantity and type of internet use in mental health, which is just getting off the ground now and is particularly well suited, I think, to the living systematic review model and the triangulation model. The other thing is that Galanos is at the moment experimenting a bit with using AI methods to essentially mine the literature and look at upcoming hot topics, which are gathering momentum, but which might not yet have even sort of consciously surfaced with professionals at meetings such as this.
Thanks. I think so that people aren't late to the plenary, I think we'll have to draw the session to a close, but it's great to see so much interest. I think the session could have gone on quite a bit longer. So thanks once again to all our speakers, and please do join our plenary tomorrow at 11:50 to learn more about Galanos. Thank you very much.